Hello, everyone. We're about 9 months into the COVID pandemic. In that time, a remarkable amount of information has been learned about the novel coronavirus and about host cell biology, immunology, epidemiology, and clinical disease. There are complexities in these subjects that confound even the experts. The purpose of this course is to learn what we know about these subjects from the world's top scientists. I'm Richard Young, and together with Facundo Batista and our teaching assistant, Lena Fayan, we are core faculty for MIT's course 700 on COVID-19, SARS-CoV-2, and the pandemic. I'm going to make a few remarks to the students who are registered for this course, and then introduce our first speaker. Students, if you miss any of the following instructions, you can find them in the course's Canvas site. First, live attendance of lectures is encouraged but not required. Recordings of the lectures will be posted with a link on the Canvas site. Your TA, Lena, will post an after lecture prompt as a survey on Canvas, a response to which will be required before the start of the next week's lecture. Answers will be graded for participation, and students need to participate in at least 90% of these prompts to pass the class. Lena will hold a session Mondays from 1 to 2 p.m., except for holidays, reviewing relevant basic material to help you better understand the next day's lecture. Recommended reading and other reading materials are posted on the Canvas site and will be suggested alongside the prompts. Students are encouraged but not required to engage with these materials. Finally, during each session, students can submit questions via the Q&A function in Zoom webinar. Selected questions will be discussed at the end of the lecture, and students are welcome to ask any unanswered questions during office hours. Our first speaker for this course is Dr. Bruce Walker. Dr. Walker is a physician scientist and immunologist, the founding director of the Reagan Institute of MGH, MIT, and Harvard, a Howard Hughes Medical Institute professor, a professor of medicine at Harvard Medical School, and a professor of the practice of medicine at MIT. He is also adjunct professor of medicine at the Nelson Mandela School of Medicine at the University of KwaZulu-Natal in Durban, South Africa, where he has catalyzed the creation of 2 important research institutes. Dr. Walker is one of my scientific heroes, a person who has played a major role in addressing the AIDS pandemic and who now is helping us address the COVID pandemic. Just last week, he and his collaborators published an important story in Nature showing that T cells in some individuals can apparently cure HIV infection. Together with Arlene Sharp, he is the co-director of the recently established Massachusetts Consortium on Pathogen Readiness, a collaboration among more than 500 local scientists focused on COVID-19 research from Harvard, MIT, BU, Tufts, UMass, and the academic teaching hospitals in Boston. Bruce, thank you so much for giving the first lecture in our course.
Well, Rick, thanks very much. I'm delighted to do this. As Rick indicated, I'm a physician scientist. I graduated from medical school in 1980, came to Mass General intending to become a family physician, but then something very odd happened, which was the emergence of a new disease. None of us knew what it was. People that had it were dying left and right. We had no treatments. And it became clear to me very quickly that if we didn't, as physicians on the front lines, learn from our patients, then we would watch a lot of people die. So I then took the route of both taking care of patients and doing scientific research, and have continued to work on HIV my entire career. And in January of this year, to put this in the perspective I have on it, I was teaching a course to MIT and Harvard undergraduates in KwaZulu-Natal, South Africa, called Evolution of an Epidemic. And this is a course that uses HIV as a model to try and understand how an epidemic emerges, how clues from patients guide scientific discovery, and how policy and advocacy influence the course of an epidemic. Now, as chance would have it, one of the things that we did was we were visiting traditional healers. And as chance would have it, one of the people in the course was an MIT student who had just returned from Wuhan, And another was the director of emerging pathogens for Gilead, a former trainee of mine who had invited along as one of the speakers. The student from Wuhan started getting text messages multiple times a day from her family talking about what a desperate situation it was there related to this new pneumonia that was emerging. And Diana Brainerd, the person from Gilead, was getting phone calls every day from physicians in China asking her to release a drug that they had developed for Ebola called Remdesivir so that they could try it to see if it had any effect on this new deadly pathogen. It became clear to me from those conversations that this had the potential to be a big problem. As Rick said, I'm the director of the Reagan Institute. Our mission is to harness the immune system to prevent and cure human disease with a focus on infectious pathogens of global importance. And our strategy is really to use cross-disciplinary collaboration coupled with flexible funding to try and make advances. And we're fortunate to be embedded in, I think, one of the best biomedical research ecosystems anywhere. So we started working on this really right away. And what I would like to do now as the first speaker is to give you a bit of an overview of what we know. How did this pandemic start? How does it cause disease? The virus, SARS-CoV-2, what are the prospects for treatment, and what are the prospects for vaccines? So getting right into this, let's put it in the perspective of what we experienced with HIV. So HIV was recognized in 1981, but the infection started being transmitted in the US, at least, as early as in 1978 or even earlier. So in 1981, it was clear that something was going on. It was 83, 2 years later, before we knew what was causing this new disease. And it took 2 more years to develop a diagnostic test so we could know actually who had it. Now let's compare what happened with this particular pandemic. So what happened was that patients started coming to hospitals in Wuhan with a pneumonia of unknown cause. And looking back at this, it was the end of December when this cluster of cases happened, and there was a really important observation made by healthcare workers, and that was that these people had a common experience of having been at a Hunan market, suggesting that this might be a transmissible agent. That led to the use of shotgun sequencing to identify the etiologic agent and to the development of a diagnostic test based on PCR. That all, which took 4 years for HIV, occurred in less than a month. So an extraordinary tribute to the advances that have been made in science over the years. The sequencing allowed scientists to recognize this as a beta coronavirus, which is an RNA virus that has a single strand of RNA and then an outer envelope with a predominant spike protein that's a major target for the immune response. The origin of the virus is almost certainly from bats originally. It also is very closely related to a coronavirus that's been identified from pangolins, but likely the transmission went from bat to pangolin, back to bat, and then to humans, although the epidemiology is still not certain on that score. This is not the first pathogenic human coronavirus that we've seen. SARS in 2003 caused a total of 8,000 cases with a fatality rate of 11%, but thankfully, the transmissibility of SARS was not that high. MERS came along in 2011 and still persists. There have been 2,500 some cases with a much higher case fatality rate of 34%, but again, thankfully, low transmissibility. But look at the contrast to SARS-CoV-2. We now have 25 million, over 25 million cases as of this morning. The estimated fatality rate is somewhere between 0.5% and 1%, but varies widely depending upon the age group and the geography, and it's very transmissible. I think the important thing for everybody to recognize from this slide is that we can expect to have additional coronaviruses in our future. And the big fear, and I think what's an existential threat to humanity, is the possibility of a coronavirus with the transmissibility of SARS-CoV-2 and the case fatality rate of MERS. So it really does behoove us to monitor for these emerging pathogens. Why is it that SARS-CoV-2 is spread so effectively? Well, it's in large part because the transmissibility begins before the onset of symptoms. And in fact, this is very different than MERS and SARS, which only are at peak transmissibility a number of days after onset of symptoms. This makes it very hard to do case tracking and isolation of infectious individuals. Not surprisingly, we have a huge pandemic, 25 million global cases, 850,000 deaths, and over 6 million infections in the US, which is the most affected country in the world. So how is it that SARS-CoV-2 causes disease? I'd like, because of the diverse audience, to start out with just a basic concept of understanding what's the difference between a virus and a bacterium. Looking here at E. coli compared in size to SARS-CoV-2, I've exaggerated the size of the virus here compared to the bacterium, but there are significant differences between these two. E. coli has over 4,000 genes. It, like other bacteria, is metabolically active, and it's capable of independent reproduction. SARS-CoV-2, on the other hand, has a total of 10 genes and 14 open reading frames. It's metabolically inert, and it cannot reproduce independently. Instead, it depends on the ability to infect the human host and to commandeer the human host to actually make the genes that are normally making proteins for the cell to use for its own purposes, to hijack that and force the cell to make viral proteins so that the virus can replicate and make new copies of itself. When the initial infections were identified to be in the lungs, that immediately focused clinicians on obtaining specimens and trying to find the etiologic agent, which is what happened through sequencing of lung epithelial cells. And that also led to a search for understanding how the virus was actually gaining entry into those cells. And that led to the identification of ACE2 as the receptor. There was a head start on this because this is the same receptor that SARS uses, the original SARS coronavirus. That is, however, those are not the only cells that become infected. Goblet cells and ciliated cells in the nose are quite infectable, and that actually probably facilitates transmission. Lung pneumocytes are infectable. That's how we get pneumonia. The gut is also infectable, resulting in diarrhea in some individuals. The virus can also infect endothelial cells in the circulation, and this is thought to lead to a coagulopathy and clotting that results in a great amount of morbidity. Cardiac myocytes can also express the receptor for this coronavirus, and myocarditis has been another disease that's been noted, and it's even appears possible that the central nervous system cells can also be infected, such as olfactory neurons. This infection doesn't happen without the body taking notice. And one of the ways that the body fights back is that B cells, one arm of the immune response, start to make antibodies, which are proteins that directly bind to the virus. They're generated in response to the virus. They learn in lymph nodes how to recognize it effectively, and then they target it and destroy it. If, however, cells get infected, then another arm of the immune system kicks in, so-called killer cells or cytotoxic T cells. These recognize infected cells because once a cell's infected, it alerts the body to the fact that something bad is going on inside that cell, by presenting viral peptides at the cell surface in conjunction with an HLA class 1 molecule, a surface receptor on these cells. Foreign peptide in the HLA molecule in the binding groove alerts these killer cells to the fact that something bad is going on. They recognize that and deliver a lethal hit to the cell, killing it, and thereby eliminating infectious virus. So we have these immune responses that are being generated to this virus the same way that they are to other viruses. Why is it that they're not doing a better job? Well, it turns out that B cell antibody production is impaired, and this is work that's from Shiv Pillai, who will be speaking later in this course, where he noted through autopsy studies that in fact, unlike a normal immune response that gets generated within a lymph node where these germinal centers result in antibody maturation, affinity maturation that allows for recognition, what happens in COVID-19 disease is that these germinal centers don't form. So there's an abnormal immune response underway that we're all attempting to understand better. Let me introduce now a second concept, and that is to make sure that we understand the difference between infection and inflammation. Infection is invasion of the body by disease-causing agents, their multiplication in the body, and then the reaction of host tissues to that pathogen. Inflammation, on the other hand, is the body's process of fighting against things that harm it, such as infections, by releasing chemicals that are also called cytokines that call the immune system into action and recruit other cells. So the second reason why the immune system doesn't do a better job is because it appears that these killer cells are ineffective at eliminating infected cells, but they do produce a lot of cytokines, these chemical messengers. In the same way, infected cells release chemical messengers. These then act on other cells and activate them to elicit even further chemical messengers. And all of this immune activation leads to bystander problems and leads to this hyperimmune state, which has been termed an inflammatory storm or a cytokine storm. And here, looking at a normal lung on the left, you can see that the alveolar air sacs, which is where gas exchange happens between taking a breath in and getting oxygen into the bloodstream, you can see that those are very fine membranes that allow for ready passage. On the right, you see what happens in COVID-19 disease. There's this massive infiltration of inflammatory cells, marked thickening of the septae, and an impairment of gas exchange, leading then to the clinical problems with lung disease. Now, essentially, what I've just told you is that we have an immune response that's designed to basically generate an antiviral immune response, and that happens early on. But what we believe happens in COVID-19 disease is that is actually replaced by a host inflammatory response that actually, rather than acting as friend, actually acts more as foe in the later stages of disease as there's increasing disease severity. We know that this disease does not affect everybody equally, probably not surprisingly, because the older people get, the more likely they're going to have comorbidities that make them less resilient to diseases in general. And in fact, what was striking early on was a sense that children did not get infected, but sadly, that is not true. And in fact, in data that just came out yesterday from the American Academy of Pediatrics, there has been recently a greater increase in cases, hospitalizations, and deaths, a percentage increase in children compared to the percentage increase that we've been seeing in adults. On top of that, there's a unique inflammatory syndrome that has been recognized in children, which is similar to Kawasaki disease, which is an inflammatory disease of unclear etiology. But what you can see on this slide is that Between 2015 and 2019, before the advent of COVID-19 disease, there were sporadic cases of non-severe Kawasaki disease and occasional severe cases. But look at what happened in 2020. Suddenly, this enormous spike in infections. We think that it's due to a post-inflammatory or post-infectious inflammatory response. What exactly causes it is still being investigated, but is of critical significance related to vaccine development for children. I'd like to say a few words about diagnostics now as a key component to trying to curtail the pandemic. What you're probably most familiar with are RNA tests, which is PCR, and this is what's being done through the Broad Institute for all of the MIT and Harvard undergraduates. These RNA tests use polymerase chain reaction to amplify fragments of viral RNA. So they don't tell you whether what's being amplified is actually infectious or not. They just tell you that there's viral genetic material there. And if you look, as was done at Mass General Hospital, at RNA detection rates following admission to the hospital, what you see is that the percent of people that are admitted who start out positive declines over time, but some people remain RNA positive for 4 weeks or more. The question is, are these people infectious all that time? Because we're right now using the RNA detection assay as a yes or no assay. Well, it turns out that there's declining infectiousness over time to the point that the CDC revamped its definition of recovery to mean 10 days after symptom onset and at least 3 days of the absence of symptoms. It's really critical that we develop an assay that we still don't have that tells us, that measures the infectiousness of a person. Now, another diagnostic test that's been developed it's a category called antigen tests, where you're not looking for viral genetic material, you're looking for viral proteins, such as the membrane proteins in blue or the spike protein in brown here. And the way that these diagnostic tests are being done is very much like the point of care diagnostic test that's used as a pregnancy test, where what one does is put a detection strip into urine in the case of a pregnancy test, and in the case of and in the case of COVID-19, what you would ideally use is saliva or a nasal swab, and then use a swab that would tell you whether or not you're infected by having 1 or 2 bands on this strip light up. These are now actually available. They're less sensitive, but in fact, less sensitive may actually be better. So let's just think about the different assays I just told you about. The RNA-based tests are much more expensive, and they require really much more extensive machinery, although that also is being addressed. Rapid antigen tests are much cheaper. Sensitivity is lower, but scale-up ability is higher, easier to get to underserved areas, easier for frequent testing, easier ease of use. But at least right now, they're not quantitative. RNA tests can be quantitative, but we haven't used them that way. And I think there's a move afoot to do that. And there are supply chain issues that are predominantly for the RNA tests, but not so much for the rapid antigen tests. So another important diagnostic is to figure out who's had the infection already. And this is done by testing for antibodies, which are proteins in the blood that are generated in response to infection. When somebody gets infected, initially there's a wave of virus that occurs. The immune response responds by generating antibodies. And ideally, these antibodies then, after the infection is cleared, come down to a level and persist to protect against subsequent infection in the future. If you look at antibody responses in people who are hospitalized, the same cohort, you can see that they come up slowly over time, and within about 3 weeks, almost everybody has antibodies. So they're, sorry. So we have ways to now really track the infection, figure out who's infected. And these antibody tests are going to be a really important epidemiologic tool. Well, let's go on and talk about what are the prospects for treatment. There are two approaches that in general are being pursued. One is antiviral therapies that directly attack the virus. And an example of this is Remdesivir. Remember in the beginning, I told you Diana Brainerd was getting calls in South Africa to release a drug that had been developed for Ebola. That drug was Remdesivir, and it's now been shown to be to have some effect. Now, the important thing to note from this paper that was published in the New England Journal is that although Remdesivir does have an effect in speeding the time to recovery, there's no change in mortality that's statistically significant. So it's really a marginal benefit, and it has to be given intravenously. We need drugs that can be given orally, that can be given early in the onset of disease and or in later disease and have an effect. It probably makes a big difference when drugs are given in terms of their ability to make an effect, and I'll come back to that. Another approach is to use neutralizing SARS-CoV-2 antibodies. And these can come from convalescent plasma, which has been given an emergency use authorization and has been a subject of much controversy as to whether it really works. Another therapy on the horizon is monoclonal antibodies that are able to neutralize the virus that could be given prophylactically or potentially therapeutically, but those are not yet licensed. Another way to approach therapy is to have directed therapies to block the cytokine storm. So these are host-directed therapies, such as antibodies to interleukin-6, which is elevated. It's a pro-inflammatory cytokine and helps to create this cytokine storm. Another is dexamethasone, a steroid. Dexamethasone has been shown to have some effect and is now being used. But there are lots and lots of trials that are underway and still no clear front runner of a drug that truly meets the requirements, I think, to have an impact of being readily available, orally ingestible, rapidly acting, and active during all phases of infection. So let me move on now to talk a little bit about what the prospects for vaccines are, which is really what I think it's going to take for us to get out of this pandemic. Again, a basic concept to make sure that we're, or basic concepts to make sure that we're all on the same page. Immunity is protection from an infectious disease. If you're immune to a disease, you can be exposed to it without becoming infected, or you can have a very attenuated infection such that you don't even notice that you've been infected. Immunity can be induced by at least two different mechanisms. One is infection itself, and that hopefully leads to immunity to any subsequent encounter by generating, for example, antibodies that persist. Another is by immunization, which is the giving of a vaccine product that stimulates a person's immune system to produce immunity to a specific disease, protecting that person from disease. So, in other words, you're training the immune system to attack the virus without the person ever having seen the virus by giving just a portion of the virus in a vaccine and using that as your training vehicle. So right now, there are probably close to 200 vaccines in development. In terms of where we are in human trials, there is a plethora of vaccines that have already entered human trials. 23 vaccines are in phase 1, which is the first phase of study where you test whether vaccines are safe and you determine what sort of dosage can be tolerated. Phase 2, there are now 14 vaccines in that stage, which is an expanded expansion of the phase 1 trial to get more safety information and learn something about immunogenicity. Phase 3 trials, of which there are 9, is when large efficacy trials are begun. I think you've all heard that the goal is to determine efficacy of a vaccine as quickly as possible. And the way that that will be done is by dramatically increasing the number of individuals that receive the vaccine. So the trials that are planned are somewhere between 30 and 50,000 patients, because you have to have enough infections occurring to show that the vaccine works better than the placebo. Finally, there are 3 vaccines that have been approved for earlier limited use, not by the FDA, but by other countries, China and Russia in particular, but there are no vaccines yet approved for full use. There are 4 different general categories of vaccine that are being pursued. One is genetic vaccines. The Moderna vaccine here in Boston is one of those. They're based on messenger RNA. DNA is another form. These use 1 or more genes to stimulate an immune response, genes from SARS-CoV-2. Inactivated viral vectors is another approach that's in part being done here in Boston by Dan Baruch in collaboration with Janssen, and also by AstraZeneca in collaboration with Oxford. This is the use of another virus to deliver SARS-CoV-2 genes to cells to stimulate an immune response. A more standard approach that's been used for years is viral protein plus adjuvant. This is a little further behind in the pipeline, but it uses SARS-CoV-2 protein or protein fragment along with something to help stimulate the local milieu where the injection is made in order to stimulate an immune response. And finally, inactivated SARS-CoV-2, which is just inactivation of whole virus, which has also been used in the past and is what the Chinese have already started using in some circumstances. Let me tell you a little bit about what we know from the work that we've done here in Boston. This has been led by Dan Baruch, one of the founding members of the Reagan Institute, who's also a physician scientist and at the Beth Israel Deaconess Medical Center. As soon as the virus sequence was released, Dan ordered synthetic genes, started vaccine design, built on a collaboration already existing with Johnson & Johnson by the end of January to work together. Then tested the vaccines in animals during February, established a challenge stock, and by the end of March, already had data suggesting that this was immunogenic in monkeys, and Janssen made the decision to go forward with scaling up production in case the subsequent studies would show that they were that the vaccine was safe and effective. The first human studies are going to be done in this month, and the expectation is by early 2021, there'll be massive scale up and emergency use authorization for the vaccine. Here are the data in monkeys. On the left, sham vaccination. Red is the composite for 10 animals. You can see that there's a marked increase in virus in this case following vaccination and then challenge. But on the right, you see the marked difference using the spike protein as a vaccine antigen. And of the 4 animals that were in this, 2 of them actually never had a blip in virus, and 2 of them had a markedly attenuated level of virus in the bloodstream. So the goal here is that instead of what happens with infection, where you generate an antibody response, what will happen here is that through vaccination, you'll generate antibodies, and then if the person then encounters virus, what will happen is either minimal infection or, in fact, no infection related to a massive increase in antibodies when they encounter the true virus infection. So how are we going to know if a vaccine is effective? Well, we have to test it in a place where there's a lot of transmission. And sadly, in the world, there's still a lot of transmission occurring, most of it in Asia, but also in South America, and North America, and rising numbers in Europe. And the New York Times on the front page this morning was an article about concern in Spain that there's a recrudescence of new cases. South Africa has had a major spike, but that has dwindled, but it's with extensive curtailment of activities. Also, we know, as shown in the New York Times vaccine tracker, that we're seeing lots of cases of infection that are being detected in colleges, which may be another place to test a vaccine, although many of these are actually going more to online education. So as we move forward, we're going to have to ultimately prioritize and decide what are the vaccines that we really want to take, and what do we really want to promote to make 7 billion doses to be able to make it available for the entire world. The one question is, will the vaccine protect from infection or does it just protect from disease? And ideally, we'd like to have a vaccine that completely protects from infection. That would be an antibody-based vaccine to get free virus before it can infect cells. But even something that protects from disease and keeps virus low enough that people won't transmit would be extremely beneficial, and that would likely depend on T cells. How many doses are needed for protection? Well, if you need 3 doses given over 6 months to get to protection, that means that a vaccine available to be given in December won't actually protect people until next June. Will there be enough new infections to show it protects? I've just gone over that. How soon can the vaccine be available? And here I think one has to be aware of when the first vaccine is in a vial and can be given to a person versus when that vaccine is available worldwide and you can go into your doctor's office and he's got it on his shelf or her shelf and can pull it off and give it to you. And so that's going to require massive scale up. And I think all of us hope that multiple vaccines will actually make it to the finish line, because none of these is going to easily make it to the level of 7 billion doses. For the vaccine that Dan Baruch is making with the Reagan Institute and BIDMC, they have committed to making 1 billion doses. So the next topic is, can enough of the vaccine be made? It's going to be challenging, but people are scaling that up right now. A critically important question is, does the vaccine work regardless of age? Most of the vaccine trials are not including people that are much older. The few that have in humans have shown that people that are most susceptible in terms of age are less likely to make an immune response to the vaccine vectors that they've been given. Another critical question is whether a vaccine requires a cold chain. And this is really critical when one thinks about global delivery. If you have to keep the vaccine on dry ice and you have to deliver it to rural areas in Africa, moreover, if you have to aliquot the vaccine once you get to the community that you're trying to administer it in, that requires special facilities and special measures that will make it much, much more difficult. So ideally, we want no cold chain requirement. How durable is the immunity the vaccine induces? Well, there already have been some suggestions of reinfection of people that have already been infected. Shiv Pillai's work suggests that the antibodies being produced are sort of not normal, in that the lymph nodes themselves are not normal. They don't generate germinal centers. So I think personally, I think the word is still out, or it's still unclear whether these immune responses that are generated by natural immunity actually do provide long-term protection, and time will tell. Another critical question is, do vaccine-induced antibodies enhance infection, or is it possible even that they may contribute to multifocal inflammatory syndrome of children? And another thing that's not on this slide is that, in fact, children or people yet less than 18 years old is a whole other category of individuals that appropriately are not being tested right now in the first wave of these vaccines as we're looking for safety. But when the trials are done in people that are 18 to 65 years old, the question is, will there need to be a whole separate set of studies done in children in order to get licensure to give these things to children and know that they're safe in children? So this is a really important point that I don't think has been given enough attention. At least it's not clear from what I've read what the actual plans are for development and licensure of these vaccines for children. So I think while we're waiting for a vaccine, we have to ask, what else can we do? We, in the Boston and Cambridge area, I think as a group of scientists, recognized that this was going to be a really challenging problem, and we felt we had something to contribute. In fact, we felt we had something to contribute on a grander scale if we could get everybody collaborating together. So on March 3rd, I had, I think what for all of us was a really unique experience where we got together with about 85 scientists from Harvard, MIT, UMass, Tufts, and Boston University to talk about what we as a scientific community could do and a clinical community could do to address this pandemic. And what came out of that was the establishment of the Massachusetts Consortium on Pathogen Readiness, MassCPR, which has been an extraordinary experience for all of us involved. I've never seen the kind of collaboration that has evolved from this consortium. We have 6 different working groups in areas ranging from clinical and outcomes research to diagnostics to therapeutics to pathogenesis and to vaccines and epidemiology, and a huge amount of collaboration and sharing that's been happening. We've also been closely collaborating with investigators in China who are at the Guangzhou Institute for Respiratory Health, which has been a fabulous collaboration. And I'm really optimistic, given the number of people working on this problem and the selflessness with which people have been working, that we will make real progress going forward. And I think we already have seen extraordinary progress up to this point. I think the other thing that we can do is follow the science. We know how transmission occurs, and there's been a lot of talk about droplets versus aerosols and how long they stay in the air, et cetera. But I think the critical experiment has really been done. And the fact of the matter is, we know how to prevent transmission. And that experiment was done in the hospitals where masks were worn by everybody. Distancing couldn't actually be achieved, but hand washing could. And what we did not see was major outbreaks among healthcare workers. In fact, at Mass General, the incidence of infection among healthcare workers is less than in the community. I think that shows us that masks work. And I think while we're all working as vigorously as we can to come up with solutions in terms of therapeutics and vaccines, if we wear our masks, we will prevent transmissions from happening. So let me make a few conclusions. One is that advances in other fields have clearly accelerated responses to COVID-19. Everything that we've done at the Reagan Institute has been based on things that we learned from working on HIV, including the entire backbone for Dan's vaccine. SARS-CoV-2 infection is transmitted prior to symptoms, which is how it spread so rapidly and why it's so important that everybody wear masks. Infection results in a hyperinflammatory state involving multiple organs, and that is what we think contributes to a lot of the pathogenesis. There are currently no highly effective treatments, even though there are some that have marginal impact, but we really need better drugs. And while we're waiting for a vaccine, effective prevention can be achieved with masks, physical distancing, and hand washing. There has been unprecedented progress toward a vaccine. We have to be sure that that vaccine is safe and effective by the time we actually administer it more broadly. And clearly, there's still much to be done, and much that people, no matter what your discipline is among the students that are listening to this talk, it intersects with a pandemic, whether it be in the scientific realm, the psychological realm, the therapeutic realm, the economic realm, et cetera. So we can all contribute to a solution. And with that, I will thank you for your attention, and I'm happy to answer some questions.
Bruce, thank you so much. The students have a number of questions. One is asking you, why do you think the US is among the most highly infected nations in the world?
Well, I think that you can actually see that in the tracings that I showed. In the last month or so, the number of infections have come down in the US, and that is directly correlated with increased adherence to the use of masks and social distancing. Other countries implemented those measures right away. They did complete lockdowns like we did, but came back from the lockdowns with continued really vigorous adherence to the prevention measures that were available to us. You know, and it's not really a surprise that that masks would be important. In fact, it's important to you wear a mask so that you protect other people. I don't think anybody on this call would want to have open heart surgery with their physician, surgeon, and OR nurse not wearing masks. They don't wear them because they're afraid of getting some infection from the patient. They're wearing them to prevent infecting the patient. We know that masks can do that, and so I think that's a critical factor here, and one of the unknowns for the future as to whether people will really adhere to that, and whether people will understand that there are times when we need to limit our own freedoms in order to protect the population at large. And there are lots of examples of decisions that we as a society make to do just that. And I think this is a situation where we need to do the same.
And Tara is asking about how long after recovery from infection does the antibody test results remain positive?
So this is a really critical and an unanswered question as yet. So we only know as long as the epidemic has been around, and there is evidence that in some people, antibody levels are declining to become undetectable over a fairly short period of time. There is concern I alluded to for at least 4 cases of purported reinfection of people that have already been infected, presumably because antibodies, which we have very good evidence that the antibodies can be protective if they're there, but these people have gotten reinfected. We don't know their antibody levels to really know how that correlated, but that's a really big concern. And obviously that would have big impact for the development of herd immunity, which is when enough people have immunity that others won't, that the virus can't easily transmit through a population because it doesn't have enough, there aren't enough susceptible hosts. Now, the question that was asked was about somebody who becomes infected, how long do those antibodies last? Now, I think the data that Shiv has generated suggest why they may not last long. But what about immunization? Well, how long will those antibodies last? And that's something else we don't know. But my sense is that that's likely to last longer, because rather than trying to generate an antibody response in a lymph node that's infected with live virus, in this case, you're taking a fragment of virus, delivering it to a lymph node, and the lymph nodes learning how to, teaching the immune response to recognize it without being impaired by the ongoing virus replication. So I think that those are likely to be two very different things, but time will tell.
Len is asking if you can comment on the mutability of SARS-CoV-2 and the impact of emerging mutations on our plans to vaccinate.
Yeah, this is another really good question. So SARS-CoV-2 is nothing like HIV, which mutates at an incredible rate. In fact, if I compare an HIV virus sequence from Boston to one in South Africa, they may be 40% different in the envelope. So we've never dealt with trying to make a vaccine to a pathogen that diverse. In contrast, SARS-CoV-2 has very few mutations. It does mutate. And the consequence of those mutations on immune recognition are still being sorted out, but there is concern. And clearly the whole population, the global population of SARS has already shifted to some extent. And I know that Dan has already made another vaccine with a second strain of SARS just in case. What will be critical is whether the mutations that arise, albeit we expect them to be far, far less than with HIV, whether those will actually result in immune escape. And again, this is a reason why we need to do lots of sequencing and lots of surveillance to keep ahead of this. And it may be that like other infections like influenza, there's a requirement for updating vaccines on a regular basis.
Another student's asking for the genetic vaccine or vaccines using an inactivated viral vectors, what are the concerns of long-term effects of the vectors or delivery methods?
So with the genetic vaccines, there's less experience with those. And so the clinical trials that are ongoing now, both for other diseases, for example, Moderna has other pathogens that they've developed vaccines for, so they have more human data from those. DNA vaccines likewise have been used for other pathogens, so that data have been generated. In terms of inactivated viruses, we've used those for years. You can ask why didn't we do that approach with HIV? In fact, in the monkey model, it looked like an inactivated virus worked, but the concern is just whether the virus could somehow become reactivated and become infectious. And I think that's tempered enthusiasm for inactivated vaccines, but there are, I think, ways to make an inactivated vaccine safe, and but that's the main concern. Another concern is that it's more laborious and expensive to make inactivated vaccines because you have to grow up a huge amount of virus in order to do that. And these entirely synthetic approaches that can be used, like with mRNA, are potentially a real plus if we can do that without having to rely on cell culture and other aspects that are required for the more traditional approaches.
Many students had questions about reinfection and the possibility that reinfection is a consequence of the antibodies just not doing what they need to do. Do you have a view on reinfection, how that, the extent to which it happens,
yeah,
the implications?
So, and there's another concern about reinfection based on dengue virus, and that is whether the antibodies could actually make things worse the second time around. One of these cases of reinfection, not to alarm people, did look like it was worse the second time around. But so far, there's no evidence from the earlier SARS data that I think are compelling. There's no evidence to suggest that there's this phenomenon of antibody-dependent enhancement. I think in terms of protecting from infection, antibodies are going to have, neutralizing antibodies are going to be required to do that from reinfection. So not everybody generates sufficient levels of neutralizing antibodies probably from their first infection. I think, though, that T cells may also play a really big role in cases of reinfection, because if the virus slips through the first defense of antibodies and gets into cells, then T cells are the main effector mechanism that can eliminate virus. And so I think attention to vaccines that not only produce antibodies but also produce T cells is important. Moreover, there's a second kind of T cell besides the killer cell. Those are helper cells that help to orchestrate an effective antibody response in those germinal centers. And so generating COVID-specific T helper cell responses, I think, may also be really, really important, particularly for the prospect of potential reinfection.
Bruce, we so appreciate you giving this an introduction to the disease, the virus, the pandemic. Thank you very much. And we'll see all of you again next week.
Thanks very much. I enjoyed it.